It is my pleasure today to introduce the speaker for today's ERS seminar, Dr. Stephen Knoll. Stephen received his undergraduate degree from Geneva College in 2015, valedictorian. Then he did his PhD with Steve Giovannoni at Oregon State, and trying to determine whether it was University of Oregon or Oregon State. And then after that, he joined the thermophile research unit at the University of Waikato, also ICTAR, under the late Professor Craig Carey, working on the Magma project, which was the last Marsden project that Craig held before he passed away untimely. And Stephen has now received his own Marsden Fast Start grant, and Stephen has published more than a dozen papers since he received his PhD 4 years ago, which is a large number for people with that number of years post-PhD. And today he's going to talk to us about his current research on Mount Erebus, and that's it's going to be a fascinating ride.
Right. Yeah, you don't want to get University of Oregon State mixed up.
No, that's why I was hesitating.
All right, so thank you all for coming. I'm going to be talking today about some of the research that I have been doing and hopefully will be doing on the microbiology of Erebus. Just for context, if you notice, earlier in the video loop, there you can see little people on the ground. That's us at a Western Crater, which you'll recognize soon enough. And that there is Side Crater, and kind of to the top is the main crater of Erebus. First of all, I just want to acknowledge I shamelessly stole part of my talk title from this book, which is a very excellent book by Colin Monteith about the history and volcanology and so on of Erebus. I highly recommend it. Also, before I go further, I just want to acknowledge all the people who have helped make this research possible, from conceptualization all the way through the data analysis. So a lot of different people have helped at various stages, so I just want to acknowledge that. As Charlie mentioned, some of the research I'll be presenting on today came from the Magma project, which was a Marsden-funded project, international collaboration. So you can see some of those international collaborators there. And then my new Marsden is there in the bottom column. And I also just want to take a moment to acknowledge two members of the Magma team, Jan Amund and Craig Carey, who both passed away unexpectedly at the beginning of last year. And so I just want to acknowledge their contributions to the research I'm presenting today, and in particular Craig. Without him, I wouldn't be here today before you. So I want to acknowledge their legacy as well. Okay, so today we're going to be talking about Mount Erebus, why we're interested in Mount Erebus from a microbiological perspective, what lives there, what kinds of functions might the microbes there be doing, and why Erebus is a great setting to hunt for globally distributed thermophiles. And then I'll end with a teaser for my latest project. And what happens when there's no sunlight every year. Okay, so Mount Erebus. You probably, when you think about Antarctica, don't think about volcanoes, but there are a very few active geothermal sites in Antarctica. Mount Erebus is one of them, and in fact, it is the largest geothermally active site in Antarctica. It's the southernmost active volcano in the world. So it's located here on Ross Island. You can see from the satellite image, this is Ross Island. Erebus is here. Conveniently, Scott Base is over here. Just a quick 20-30 minute helicopter ride away from Mount Erebus, which makes it quite easy logistically in some ways to do field work on Erebus. So Erebus is unique from a volcanic and geological standpoint because of the type of volcano it is. So it's a phonolitic volcano. So that refers to its mineral composition. It is also active, so it has a permanent lava lake in the main crater that regularly erupts. Most of the lava bombs it produces do not leave the main crater, but it does regularly erupt. It is one of the few, perhaps only, phonolitic volcanoes in the world that is active, so that makes it quite unique from a volcanology standpoint. And also unique about Erebus is the fact that it's in Antarctica. So you have this intersection between freezing cold air and very hot soil and gases and steam. And so that can create some pretty cool features that we'll talk about soon. But it's not just the main crater on Erebus that has geothermal activity. So at various sites around the summit of Erebus, you can find areas that have hot soil. So this site here is called Tramway Ridge. It is the largest, one of the largest sites around the summit that has geothermal activity. It is also one of the hottest. Maximum soil temperatures you find there are 65 degrees, whereas the air temperature is, less than -20 year-round. Tramway Ridge is also unique because it has a lot of steam, as you can see from the picture, and it also has a lot of cyanobacterial mats and also a lot of mosses, which you don't find elsewhere on Erebus. And because the soils are so hot, here at Tramway Ridge, you just find bare hot soil for the most part. At other sites, like Western Crater, which you saw in the video earlier, the maximum soil temperatures are only about 50 to 55 degrees. You don't find any sorts of mosses or cyanobacteria mats. Because the soil temperatures are lower and there's not as much steam, that steam, as it is produced out of the ground, condenses and freezes. And so wherever you find a hot soil site at Western Crater, it's almost always covered by a little shell of ice or ice hummock. And so you can see those in the picture there, the little domes of ice. Now, on some sites on Erebus, you will find a lot of steam coming out of the ground in a concentrated site. The soil temperature isn't that hot. So at these sorts of sites, you get the formation of ice towers or ice chimneys. This one here in the picture is 10 to 15 meters tall. So this can be massive. And it's all, again, from that process of steam coming out of the ground, condensing and freezing over time. And eventually they get too big, they fall apart, and then they form again. So it's kind of a life cycle. And oftentimes you find these ice chimneys associated with ice caves, which are formed when a hot soil site starts up from volcanic heat in the ground underneath the ice pack, and it melts away the ice and the snow until you get the formation of a cave. Some of these caves can be kilometers long, so they can be pretty massive. But the maximum soil temperatures you find in these caves tend to be pretty low, so 10 to 20 degrees Celsius. Okay, so as a microbiologist, of course, my first question is, whenever I see a place in Antarctica that has water and heat, is what lives there? And so in the early years, the microbiology of Mount Erebus was all culture-based. I just want to point out the very first paper on the microbiology of Mount Erebus was from a couple of guys that happened to be in Antarctica and thought they'd pop up to Erebus and grab some soil samples and plate them on a plate. And they got a paper in Science out of it. I wish it was that easy these days, but anyway. So these papers are all culture-based or observation-based, and they're almost all concentrated at Tramway Ridge. And they don't tell us a whole lot about what's there, other than that there are some thermophiles that live there and some cyanobacteria and moss. And then Craig came along in the 2000s, and he brought some up-to-date genetic methods like amplicon sequencing using 16S ribosomal RNA gene as a marker to look at the microbial communities present on Erebus. And so these papers in 2009 and 2014 looked at the microbial communities at Tramway Ridge, and they found pretty unique microbial community there, and in particular some pretty unique archaea, which we will come back to later on. There's also a couple of papers published on the microbial communities in some of the ice caves on Erebus. But overall, there's still a lot more to learn about what lives at some of these sites. And so we wanted to expand our knowledge of the microbiology of Erebus beyond just Tramway Ridge and a couple of ice caves, and get more of a comprehensive picture of what lives up there. So in total, we collected 48 samples. I say we, past people had collected we had 48 samples sitting in the freezer. And DNA was extracted, and we sequenced the 16S ribosomal RNA gene to look at prokaryotic diversity, as well as the 18S gene for eukaryotes, and then we did ITS sequencing as well to look at fungal diversity. We divided these samples into exposed or subglacial categories. Subglacial refers to ice caves, and exposed refers to places like Tramway Ridge that are exposed, sometimes potentially covered in ice hummocks, but for the most part exposed. We wanted to answer three basic questions. What's there? Are there differences between different sites or different types of sites? And do we see any impact of the physicochemistry of those samples on the microbial community present? And so just at a first glance, looking at the chemistry of these samples, we saw pretty quickly that Tramway Ridge is actually pretty unique compared to everywhere else on Erebus. So we split it out into its own category. So we've got Tramway Ridge, other exposed soils, and then subglacial or ice cave samples here. You can see that Tramway Ridge has, the soils on Tramway Ridge are significantly warmer, have significantly lower pH, higher conductivity, higher total carbon, higher total nitrogen, higher water content compared to everywhere else on Erebus. So right away, we can see that the past studies that were just focused at Tramway are missing a lot of the diversity that is found on Erebus. But also, Tramway Ridge is a pretty unique ecosystem in and of itself. So when we look at the microbial community that's there, I'm going to focus in my talk just on the prokaryotic results for the sake of time. If you're interested in what kinds of eukaryotes are on Erebus, feel free to read the paper or ask me afterwards. But when we look at the prokaryotic microbial communities in these sites, so if you haven't seen a PCA plot before, which I'm sure you have, each point here.
No, I'm only seeing your cursor on, at least on the projector.
Oh, sorry. Yeah, there we go. Thanks. So each point here refers to the microbial community from a single sample. The closer two samples are to each other, the more similar those communities are to each other. So the ice cave communities are all pretty similar to each other compared to all of the exposed samples. The Tramway Ridge samples, some of them are clustering kind of with the other exposed samples, but these ones up here are quite different. And as it turns out, all of these samples up here are from soils that have temperatures of 60 degrees or greater. So these are all the high temperature Tramway Ridge samples, and they're all quite unique compared to everything else in the Erebus. So the takeaway here is that we do see some differences between different types of sites, especially the caves and especially the high temperature samples at Tramway Ridge. Okay, but what's actually there? This plot probably doesn't really help that much. So each sample here gives you the relative abundance at the phylum level in that sample. And it's kind of messy and hard to look at. So we did some statistics to tell us what phyla are useful for distinguishing between different categories of sites. So for instance, for Tramway Ridge, we can see that they tend to be enriched in species from Bacteroidota, Crinarchaeota, now Thermoproteota, Dinococcoda as well, and Armidotomodota. So these are the phyla that tend to be uniquely found at Tramway Ridge. And this makes some sense, or it fits with what we already knew about Tramway Ridge. Like we already knew there are a lot of interesting archaea at Tramway. We also know that Tramway has a lot of mosses and cyanobacterial mats that we don't find elsewhere in Erebus. And we know that Bacteroidetes and Dinococcoda and Armidotomodota, those tend to be thermophilic in some cases, heterotrophs. They're going to be chowing down on all of the photosynthetically produced sugars that we would tend to find at Tramway Ridge. In terms of the ice caves, we found them enriched in chloroflexi, which really doesn't tell us very much because chloroflexi is a very diverse phylum. But interestingly, we also found ice caves enriched in actinobacteria and acidobacteria. And yeah, I'll come back to the actinobacteria a little bit later because that's kind of an interesting one. And then the other exposed soils, they're not really that distinguishable except by the presence of a lot more firmicutes like Geobacillus, which is the bane of my existence when it comes to filtration. I'll come back to that later as well. Okay, so we've got a decent idea what kinds of prokaryotes we find in these samples. But what about chemistry? Do we see any influence of the physicochemistry of the sample on the microbial community? This plot is similar, distance-based redundancy analysis. The arrows, the direction of the arrows relates to the correlation, what samples they're correlating with. The length of the arrow tells you how strong that correlation is. We can see, as we would expect, Tramway Ridge, the communities there tend to be correlated with higher conductivity, water content, carbon and nitrogen, and temperature, although temperature wasn't as strong of a factor as we thought it might be. This all makes sense with what we know about Tramway Ridge being a higher productivity site. We did see a correlation between these three elements, magnesium, calcium, and strontium. And in particular, they actually tend to be correlated positively with actinobacteria and the ice caves. And at first we thought that that was because the actinobacteria there are sporulating species. So a lot of actinobacteria make spores. And oftentimes that process requires ions like calcium and magnesium. But none of the species that we found in our data set are known to be sporulating. So we have no idea why these actinobacteria are found in the ice caves. Anyway, maybe there'll be someone down the line will figure that out. Okay, so we've talked about what lives there, the impact of chemistry, potentially on what lives there, differences between sites. But what are they actually doing? So so far I've just been talking about taxonomy based on the 16S marker gene. Taxonomy does not always correlate with function, as we probably well know. And so to try to get at potential functions in the community, we did metagenomics. So you take your soil sample, you extract all the DNA from it, you sequence all of the DNA there, and then you try to stitch back together genomes out of that DNA that you sequence to hopefully get metagenome assembled genomes, or MAGs. And then you can predict functions based on the presence of different metabolic genes in those MAGs. And so far, we have 2 metagenomes from Erebus, one from Tramway and one from Western Crater. We also have metagenomes from two other geothermal sites in Antarctica, Mount Rittman and Mount Melbourne, but I won't be talking about those here. So one helpful way to look at the metagenome data to get an idea of what the community might be doing is to look for the presence of marker genes for different types of metabolisms. And so that's what this figure here is showing, arranged into different groups of metabolic groups. So one of the things to, so the colors here correspond to the estimated abundance in community. So if, so for instance, these colors here that are 100%, you would say that 100% of the microbes in that sample are capable, have the cytochrome AA3 oxidase, which is involved in aerobic respiration. So that makes sense that everyone there would have it. A couple of things to note. First off is that Tramley, compared to Western Crater, Tramley Ridge is enriched in genes relating to phototrophy, which makes sense given they have a lot more cyanobacteria and mosses there. And also a lot more genes related to nitrogen cycling, so nitrate and nitrite metabolism in particular, which makes sense given that there's more total nitrogen at Tramley Ridge. So this kind of matches up with what we'd expect. We would expect to see a lot of kind of phototrophy and associated heterotrophy going on, especially at Tramley Ridge. But also of note is the presence of genes for hydrogen metabolism and carbon monoxide oxidation. So we know that the volcanic gases on Erebus, including at Tramley Ridge, are enriched in gases like hydrogen, carbon monoxide, and carbon dioxide compared to the atmosphere. So this makes sense that you would see a lot of microbes taking advantage of those higher gas concentrations. Hydrogen and carbon monoxide in particular are quite nice for microbes to munch on for energy. So that all makes sense. So we get an idea just based on these two metagenomes that there's a fair bit of phototrophy, heterotrophy, but also chemolithotrophy going on at these sites. And we hope to expand this to beyond just a couple of metagenomes to get a better picture of what's going on there. And Seeger, who's sitting in the back, will be working on that project, so you can ask him later about that. So I mentioned a couple of times about the interesting archaea that live at Tramley Ridge that you don't really find anywhere else on Erebus. And so we did, using our metagenome from Tramley Ridge, we were able to assemble a mag that corresponded to this unique archaea. And as it turns out, it falls within the now called Thermoproteota phylum within the Nitrosospheria class. It's quite deep branching compared to other members of the class. And this group tends to be known for their ability to oxidize ammonia. So when you hear ammonia oxidizing archaea talked about in the literature, they're almost always from this group. As it turns out, this archaeon from Tramley Ridge does not have the genes for ammonia oxidation. So that makes it quite unique. There's only been a few other species found in the world that from this group that do not oxidize ammonia. Based on the mag that we pulled from Tramley Ridge, we think they might be involved in amino acid degradation, potentially anaerobically. They also do have a gene for oxidizing carbon monoxide, but it seems to be the type that operates really slowly, so we're not sure if they're actually using carbon monoxide as a primary energy source. But until we have it in culture, we can't really say for sure what it's actually doing. But that probably won't happen anytime soon because the predicted maximal growth rate of this mag was about a doubling time, like 25 to 30 days. So we're probably not going to be culturing that anytime soon because it would take years to actually get visible biomass. But anyway, for now at least, we have this mag to go off of. Okay, so I've talked a bit about what lives on Erebus and what they're doing. And now I'm going to be talking about Erebus as a place to look for cosmopolitan thermophiles. So if you remember this paper from 2014, some of the primary findings of that paper was that a lot of the microbes on Erebus, especially Tremley Ridge, are quite unique, potentially endemic to Tremley Ridge. But also, they found a fair number of microbes that were identical, based on the 16S sequence, to microbes you find elsewhere in the world. And this may not strike you as surprising, but it actually is surprising but based on what we know about thermophiles in general. So this paper is one example of that. So in 2022, this guy took a bunch of genomes from different environments, searched them against databases elsewhere in the world to see if he could find the same species elsewhere in the world from different habitats. The way to interpret this plot is any given point in this plot on the x-axis is the average nucleotide difference between the query and the subject genomes, and the y-axis is the distance between those two, where those two genomes were sampled from. So for human, and the color corresponds to the probability of finding that genome at that the genome at that average nucleotide difference at that given geographical distance. So the interpretation here is for human microbes, you're going to find the same species basically anywhere else in the world, or at least, human microbes tend to disperse pretty far around the world, which makes sense because humans travel a lot around the world. But if you look at microbes from hot springs, they don't really travel very far, or at least they're not identified elsewhere in the world. Another way of thinking about it is to calculate the diffusivity of microbes based on this data. And the interpretation of this would be that after 100 years, you would expect a human-associated microbe to have traveled 400 to 500 kilometers, whereas you would expect a microbe from a hot spring to have traveled about 10 kilometers after 100 years. So it is generally thought that thermophiles do not travel very far through the atmosphere, which I guess makes sense because they're thermophiles, so they would want to grow in warm environments. And if you're going to be in the atmosphere, it's going to be cold, lots of UV and freezing and desiccation, starvation, all those things. But when we look on Erebus, we do find a fair number of species that are identical to species elsewhere in the world. So what gives? And actually, if you think about it, Erebus is a really great place to look for these types of globally distributed thermophiles, because if anything is going to arrive on Erebus from elsewhere in the world, it has to get there via the atmosphere. So humans don't travel to Erebus very often. And so it makes a pretty good place to look for globally distributed thermophilic microbes. So we wanted to start by expanding the 2014 data, because that was just based on 16S sequence. So we want to expand that to MAGs. So we have our metagenomes from Erebus. So we wanted to look to see if we could find the same MAGs on Erebus elsewhere in the world. And we did this using a program called Branchwater, which breaks MAGs down into little chunks and searches for them. And we use a threshold here that corresponds roughly to average nucleotide identity of 97%. So the species cutoff is 95%. So 97% is a very conservative estimate. So we can be pretty sure that if we do find a hit elsewhere in the world from our Erebus mag, that it is actually the same species. So we used our two metagenomes from Tremley Western Crater, also our metagenomes from Rittman and Melbourne, the other two Antarctic geothermal sites. And we found that when we filtered to just the thermophilic mags, based on predicted optimal growth temperature. We found that a little over half of the mags at these sites are cosmopolitan, as in they have a hit on a metagenome somewhere else in the world outside of Antarctica. And I say cosmopolitan versus endemic here. I should note that endemic doesn't necessarily mean endemic. It just means we didn't find them in any metagenomes that have been sequenced yet. I just say endemic for shorthand. And these mags that are showing up as cosmopolitan, they're not just coming from 1 or 2 samples in a few places. They're actually quite broadly distributed globally. Some of them have hits all over the world. Some of them are a bit more localized in terms of where you find them. You tend to find them coming from North America, South America, and subantarctic islands, which makes sense given what we know about where dust particles end up, where dust particles in Antarctica tend to come from. They tend to come from South America and North America. And they're coming from all sorts of different habitats, so hydrothermal vents, soils, estuaries. So these cosmopolitan microbes are quite broadly distributed. And so okay, so this is just based on our metagenomes that we have. We wanted to expand this to get some more data. So if you remember, the paper I mentioned earlier was looking at hot spring microbes. And Erebus doesn't have any hot springs, it's all hot soils. And actually globally, the number of samples, or globally, hot soils are underrepresented in sample databases. So in order to confirm our results and also add to the what is known about hot soil microbes, we're in the process of collecting and sequencing samples from a variety of different sites with a focus on North America and subantarctic islands. The goal is to do DNA sequencing on these samples, figure out what kinds of microbes live there, and if we find shared microbes between these sites and our Antarctic geothermal sites, to give more correlation, more supporting evidence that there are actually globally distributed thermophiles. That's still a work in progress. Okay, so we have these potentially cosmopolitan thermophiles. We have mags, so we can predict genes from them. So we can ask questions like, are there any defining traits of cosmopolitan microbes versus potentially endemic microbes? And so just looking at kind of some basic features, we found that the cosmopolitan thermophiles have significantly higher GC content compared to endemics. So that makes some sense. So higher GC content gives you more stability in your genome, better nucleotide pairing. So it makes sense that if you're going to be traveling through the atmosphere, you need to have some more stability in your nucleotides in your genome. The only other significant difference we found between the two groups was in the predicted maximal growth rate. Now, predicted maximal growth rate is not a very precise number, so take this with a grain of salt, but we do see a significant difference here. We do see that cosmopolitan microbes tend to be faster growing than endemic microbes. This makes sense when you think about, for instance, if you clear all the grass off of a portion of your lawn, the things that grow there first are going to be the weeds that grow really fast. So it's well known in the bacterial world, the same thing tends to be true for mesophiles. That faster growing microbes tend to be more broadly distributed than slower growing ones. But it seems like that's also the case for thermophilic microbes as well. We also had a look for traits that might be associated with long-range atmospheric dispersal, like sporulation. We didn't see any difference between the two groups in terms of ability to sporulate. But in terms of UV resistance, so these are genes that are involved in DNA repair, those sorts of traits that will be really helpful for UV resistance. We did find that cosmopolitan microbes were enriched in genes relating to UV resistance. So that makes some sense based on what we would expect. Now, if you remember, you probably didn't notice, but from our list of cosmopolitan thermophiles, we had 3 mags that came from the Dinococcoda phylum. Now, I've spent a fair amount of time over the last few years trying to culture stuff from Erebus. And as it turns out, we've managed to culture all 3 of these mags. So we've matched their genomes, the mag to the genomes of these isolates at 99% similarity. So we're pretty confident that the 3 isolates we have correspond to these 3 mags. And so now we can go do some testing in the lab to say, can these microbes actually survive the sorts of conditions that we would expect them to see during long-range atmospheric dispersal? It's also handy that we see a bit of a difference in terms of their distribution. So for instance, this the Calidothermus is quite broadly distributed globally, whereas the Alamiothermus and Thermus are a lot more narrow in their range in terms of where they're found. And we do also see differences between them in terms of their GC content that kind of line up with what we're already seeing. So it may be that we will be able to see that reflected in the physiological experiments that we're doing. So currently I'm in the process of subjecting these bacteria to freezing, starvation, desiccation, and UV. Lovely things. I spend my time destroying bacteria. And I haven't gotten all the results yet. In fact, the only good result I've had so far is from one isolate for UV. So this is the Thermus species, which is the one that was only found in one other place in the world, it's a subantarctic island. And it doesn't handle UV very well, especially when compared to some isolates from another paper that were exposed to the same sort of treatment that were a lot better at handling UV. So this actually is not too unsurprising, given that this is this isolate, the species, that's only found in one other place in the world. So it makes sense that it's not as good at surviving these sorts of exposures. But stay tuned. Hopefully we'll have some fun results in the next few months if I can get them to work properly. Okay, so we've spent a lot of time talking about what lives on Erebus and kind of what endemic and cosmopolitan microbes. And I want to spend the last 10 or so minutes talking about my new project. And of course, all the pictures I've shown so far from Erebus, the sun is shining and the skies are blue. That's because we only do field work on Erebus when it's summer. We can't go up to Erebus in the winter. And every year in the winter on Erebus, there are 3 months of darkness when the sun does not rise at all. And an extra month on the other side when there's hardly any sunlight at all. So what happens when the sun goes away? And in particular, if you remember when I was talking about Tremley Ridge, I mentioned there are a lot of phototrophs that live there. So how do these phototrophs survive every year for 3 months without any sunlight? And in particular, these cyanobacterial mats have a lot of species like Leptolingia and Fissurella or Masticocladus, and a lot of mosses as well. So what are they doing over the winter? And that's the broad question that I hope to answer with my new project. So we're going to be looking at 3 main questions. First question is, do they survive the winter darkness? So it is potentially feasible that every year all the cyanobacteria in these mats die and survive at cooler sites and then repopulate the warmer sites in the spring. So that is a possibility. So do they even survive the darkness the winter darkness every year at the warm sites. And I will just back up a minute. These mats tend to be at soil temperatures of 20 to 50 degrees. So it's pretty hot for something to just go dormant for 3 months every year. So are they surviving at the warm sites? And if so, how are they surviving? So that's our first question. Question 2 is, are the answers to question 1 different for microbes in different environments on Erebus? So some species prefer high temperatures, like 40 to 50 degrees. Some are more medium, like 20 to 30 degrees. Some are lower temperatures, like in the ice caves that have sunlight, some like 10 to 20 degrees. So the 2 different photosynthesizers have different strategies, depending on what temperature they like to grow at. And then finally, how do these survival strategies, if any, influence the broader community around them? So basically, what happens in the microbial communities on Erebus every winter, not just the photosynthesizers? Our null hypothesis is that they don't survive the winter, and there's a paper back in the 1990s where they isolated one of the Fisherella or Mastigocladus cyanobacteria from Tremley Ridge, and they did a bunch of things to it, including putting it at constant darkness at different temperatures, and they found that at temperatures of 50 degrees, which is their optimal growth temperature, they couldn't survive for more than a month without any light. And so that's where our null hypothesis comes from. And so we plan to test 3 different species from Erebus, so 2 from Tremley Ridge, the Fisherella from the high temperature zone, Leptolingbia from the medium, and then algae like Coccomixa from some of the ice caves that have light coming into them. And our plan is to get cultures of all 3 of these different species or groups, and then put them through a lab-based winter simulation. So do the same sorts of experiments that the 1990 paper did, but actually testing a few different hypotheses. So Susie and her new PhD student, Sithamini, are going to be heading up the isolation and characterization part of this project. They're down at Lincoln University. And once we have the isolates, we're going to be using our winter simulation in the lab to test for a few different hypotheses. So these are hypotheses for how they might be surviving the winter. So potentially the cyanobacteria are switching to heterotrophy over the winter, which we'll be testing just by adding different carbon sources to the media, see if that helps them survive 3 months without light. The second hypothesis is based on the presence in some of these cyanobacteria of genes for far red photosynthesis, so using far red irradiance for photosynthesis, so above 700 nanometers. And there have been papers published on some desert cyanobacteria that have these genes that use the increase in far red light every day, so as the sun is setting, as a trigger to store inorganic carbon for the next day. And so one possibility is that these cyanos are using the increased far red light in the autumn as the sun is starting to set as a trigger to start storing up nutrients for the winter. So sort of like a bear getting fat before the winter comes along. That cyanobacteria. A third and kind of more out there possibility is that they are actually doing photosynthesis using far red irradiance that is produced from the geothermal soil that they are growing on. And they're surviving over winter because they still have that irradiance from the ground year-round, even though they don't have the sunlight. Now we know from hydrothermal vents that geothermal sites can produce irradiance in these kinds of wavelengths. We don't know if it's produced in any amount at sites like Tremley Ridge, so that's something we're going to be testing. But it is potentially feasible. So we know that at some of these hydrothermal vents, so deep in the ocean, there are obligate photosynthesizers that live there. that rely on the irradiance from the hydrothermal vents for photosynthesis. So those are 3 hypotheses we're going to be testing using our lab-based simulations. And then we hope to validate them in the field using a shading experiment. So Dawn Sumner, who's our collaborator at UC Davis, has been helping us with the design of these shading experiments, since she's done similar ones in some lakes in Antarctica. And Sigurd has been very helpful in designing these shades. So our plan is to install some shades at Tremley Ridge. We're going to have 3 high temperature mats and 3 lower temperature mats at Tremley Ridge to essentially simulate winter darkness over these mats and see what happens to the cyanobacteria there, but also the broader community there. And we leave in about 2.5 weeks to do this, so it's coming up quickly. And then the plan is to return 3 months later at the end of the summer, take the shades off, do some sampling, and see what happened to the community. And Sigurd has developed these wonderful shades. If you want to know what that black part of the shade is, it's a very interesting story. You can ask me at the end about that. Okay, so we've talked about these different questions. We've talked about how we're going to address them, focusing mainly so far at Tremley Ridge. When it comes to the low temperature end of that in the ice caves, so the well-lit ice caves, we're not able to do shading experiments due to logistical other issues. And so we're planning to answer the 3rd question for the low temperature photosynthesizers using a different strategy, using an ice core that was collected over a decade ago by Craig and Ian in Hutt Caves. This is a cave that has a fair amount of light penetration into it. We know that there are photosynthetic microbes that live there, mainly algae. And so the hope is that by drilling a core through the side of the cave in an area that has a lot of light, that will serve as a sort of history of what the microbial community in the soil of that cave looks like over time. And the idea is that we'll be able to identify the winter and summer sections in the ice core, divide up the ice core into those sections, do DNA sequencing, and look at how the community has changed. And so the idea is that the soil particles from the base of the cave have been deposited into the ice over time. You can see the core being collected here, where there's a lot of those soil particles on the ice. And hopefully, this core has a sort of history that we can use to figure out what the community looks like in the summer versus winter in the ice caves. And Manon, who's also in the back, will be heading up that analysis as part of her master's. And we have Holly Winton down at Victoria University, who's a specialist on ice core analysis, who's going to be helping with that as well. Okay, so we've covered everything that I hope to cover, and I haven't even gone over time, so I'm very glad. So with that, I'll take any questions you might have.
Are there any questions online, Joshua?
I will check. Will you check? Are there any questions in the room?
Yes. So at one point you were talking about that study that quantified the dispersal in terms of diffusion coefficient, which implies it's sort of like some kind of smooth, continually happening. How does it actually, is dispersal just, some kind of storm whips something over and it survives and lands somewhere else? How does dispersal actually occur?
Yeah, it's a great question. And it's a very hard to answer question. The thought is, so when it comes to thermophiles in particular, our current working thought is that, because obviously they need to get up into the atmosphere somehow, right? And one way that can happen is through volcanic eruptions. So there was a paper published a while back that found that after volcanic eruption, that diatoms around in the area around the volcano ended up far away because of the eruption. Whether they're able to survive that is another question, but that is one possibility. Another possibility is steam. So obviously microbes getting caught up in the steam. Don't know if that'll be enough to get them very high into the atmosphere, but all you need is some wind to catch a few and get them higher up. But yeah, it's an open question. Yeah, and really like it only takes a few microbe cells surviving to start a new colony. So yeah, who knows?
More questions? Any questions online, Joshua? No, Georgia.
So with the most common photosynthesizers that you're hoping to look at, is it known what light-sensitive proteins they have, and so what wavelengths they respond to?
Yeah. So we don't have them in culture yet, so we can't answer that. But we know that at least for the Fissurella and the Leptolingia, they have the genes necessary for doing far-red photosynthesis. In addition to their normal photosynthetic genes, we would need to experimentally validate that. In terms of other light-sensitive proteins, I'm not sure. I haven't looked specifically. But yeah, that will be part of the project.
So I have a question. Well, it's more of a comment because Georgia's question has prompted you to give the answer, which is not the one I wanted, which is that because you haven't looked, right? But for one of your hypotheses, I forget which number, it would essentially require that these cyanobacteria evolve extensive accessory pigments to collect light, right? Like the photosynthetic organisms and hydrogen vents do. And so that'll be another thing for you to look into the genome.
Yeah, for sure.
You do have one question online coming from Roberto. Could you tell us more about why you chose to name the hot soil sample Los Homers? Mexico?
Did that come through? Oh, here it is.
Yeah.
Oh, yeah. So, that's the site where we have samples from, Los Tumeros. So we have a collaborator there in Mexico who's sampled before. That's a geothermal area, and they've done some sampling from that site before. So I contacted them about potentially sharing some samples with us, and they did. So it's from that site. That's why I named it that way. Yeah.
Okay. There are no other questions. Thank you, Stephen, for a great seminar.